How double helical structure replicates protein. How were protein molecules first replicated? How were the double helical structure of amino acids formed? The birth of life was first caused by the production of amino acids in a random process. The amino acids form right-handed L-type and left-handed D-type helical structures by asymmetric carbons. Those tend to form molecular pairs with a mirror-image relationship. On the other hand, molecules of amino acids of the same chirality tend to form peptide bonds with each other. Therefore, the synthesis of right-handed and left-handed proteins proceeds at the same time. In this way, amino acids made proteins. The protein useful for living organisms remain in the living organisms and acquire a mechanism replication. Replication of amino acid sequence by double helix structure. As shown in Fig. 1, the double helical structure has a mirror-image relationship because the molecules of one amino acid in the pair face each other in a way that warps back against the molecules of the other helical structure. In a mirror-image relationship, amino acids of one chirality can use the amino acids of the other chirality as a template to replicate amino acids of the other chirality. The progress of the synthesis reaction of molecular groups with different chirality is crossed. So, a pair of amino acids with a double helical structure is added at the point of intersection. Amino acid molecules have side chains R groups, which attach to the hydrocarbon molecular layer of the cell membrane. Due to the chirality of the attached amino acids, intermediaries such as cell membranes are also affected by chirality. The tissue of the molecule changed by the influence of amino acids may become an intermediary for the replication of proteins. How did the double helix DNA form? Chirality of amino acid strings and chirality of DNA. The generated L-type and D-type protein molecules have the same size of the molecular separator of amino acids, so a unit of double helix structure is produced in the same amino acid unit. As shown in right side of Fig. 2, L-type amino acid molecules are attached to L-type reading strands, and D-type amino acid molecules are attached to D-type lagging strands. The L-type and D-type bases of the compensating base pairs that bind them possesses the information of protein replication in amino acid units. In DNA, individual molecules of amino acids are represented by a combination of 3 complementary base pairs. How had been DNA formed? The replication of large proteins by tissue pairs of molecules in the cell membrane is complicated. The molecular structure of the cell membrane region and the DNA base pair is different, and the relationship is unclear at the present. It is possible that the region of the cell membrane pair, which has a replication function, has been replaced by a complementary base pair as a region with a DNA replication function. The role of cell membranes in the birth of life. Where had hydrocarbons of the cell membrane produced? The main components of the cell membranes of all living organisms on Earth are long-chain hydrocarbon molecules of C16H34 and C18H38. This material was formed in the early ancient period when hydrogen ions of the solar wind collided with the Earth's atmosphere, which was mainly composed of CO or CO2 at a high speed of about 500 km per second. Light molecules such as methane CH4 remained in the sky, and further synthesis reactions continued. Among these molecules, long-chain hydrocarbons were formed by repulsion between hydrogen ions. The molecules of C16H34 and C18H38 were liquids in the temperature range of the Earth's surface at the time and suspended on the surface of the water to form a hydrophobic film. Permeability of chiral molecule in the phospholipid double-layer membranes. Helical molecules that invade the bilayer cell membrane can pass in one direction because the input direction is reversed in the center and enters the opposite membrane, as shown at the top of Fig. 4. However, when a fatty acid with an oxygen atom enters the end, the molecule of the fatty acid stays in the membrane and forms a cell membrane. On the other hand, as shown in the lower part of Fig. 4, helical-like molecules that invade from the front and back sides of the bilayer membrane in different directions do not mix, but push each other in the center, will form a double helix structure like DNA. Thermal motion of atoms in helical structure. A helical arrangement of distorted tetrahedral water molecules may occur. In liquid water, water molecules are connected by hydrogen bonds in contact with the cell membrane, and a wide variety of atomic configurations change instantaneously in the -12th power of 10 sec. As shown in Fig. 5, the thermal motion of liquid water has a rotational thermal oscillation of tetrahedrons in helical arrangement. The tetrahedron of liquid water molecules is distorted and splits the energy levels. It brings the lowest energy state. Similarly, silicate SiO2 distorts is a substance made of a slightly distorted tetrahedron as a component. There are a number of lattice structures in the silicate, but the alpha quartz of the helical structure is the smallest in size and the lowest energy state. Coordinated thermal motion of atoms in a helical structure of water. In a tissue in which tetrahedral molecules are connected to form a helical structure, the movement of the tissue of atoms is carried out in alternating rotation with the electrical axis as the axis of rotation as shown in Fig. 2. In such environment, the left-handed and right-handed forms of molecules with the same helical structure may exist at the same time. Base pairs with hydrogen bond and mirror image pairs. Atomic arrangements of bases for replication of bases. Left and right molecular strings in DNA are synthesized by binding phosphate PO4 to 5'CH2 as shown in Fig.7. Therefore, the progress of lagging strand is anti-parallel to leading strand and the chirality is different. When the molecules of base attach to the leading strand, those are changed to the mirror image atomic arrangement. However, the atomic arrangement attached to the lagging chain does not change by the reversal of the inversion. Fig.7 shows the atomic arrangement of adenine and thymine of DNA base pairs. The base in the upper left of Fig.7 is face-to-face bound to the leading chain and thymine T becomes the mirror template mount. Moreover, mount becomes a compensated base pair by hydrogen bonding with adenine A in the lower left of Fig.7, adenine A becomes a mirror template Ma, and that Ma becomes a compensated base pair by hydrogen bonding with thymine T. If we focus on the atomic arrangement in which the reading and lagging chains are shifted, the atomic arrangement of thymine template mountain is a mirror image of thymine T. In addition, the adenine template of Ma is the atomic arrangement of the mirror image of adenine A. When these bonds are connected, the bonds are stabilized by a loop-like cyclic bond with from MT to A, and from A to MA, and from MA to T, and from T to MT. How did establish complementary base pairs? Complementary base pairs combined with mirror image mold. Suppose that the replication of DNA is performed by compensated base pairs with replication by mold of template. As for the replication on between cross over the row by shift of the line, other base pairs possible to be included in between the rows of the upper and lower base pairs in Fig.8. For the sake of simplicity, let us confirm the structure of Fig.8 with complementary base pairs as follows. Condition 1. Thymine template mount must be a compensatory base pair with adenine AIE, molecule 2 is the compensating base of 3. Condition 2. Product of the thymine mold mount is thymine TIE. The molecule in 4 uses the molecule 3 as a template. Condition 3. Adenine A is a product of the adenine template Ma, i.e. the molecule in 2 is the molecule in 1 as a template. DNA is replicated by satisfaction of the conditions of 2 and 3 under condition 1. Then generation of the next base pair will be performed. As shown in the right side of Fig.8, the same operation is performed to compensate for replication conditions for guanine and cytosine. How to synthesize amino acid sequences for protein. DNA with 2 complementary base pairs of AT and GC. The Watson and Crick DNA model is shown in Fig.9. Since amino acid molecules are bound to the outside of the cell membrane and cause structural changes in the cell membrane, they can be linked to the combinatorial unit of the 3 base pairs of DNA. The left and right molecular sequences of DNA are horizontally connected by 2 sets of base pairs, adenine A and thymine T and guanine G and cytosine C. Therefore, if we allow superposition from 4 combinations in which the order of the 2 base pairs is swapped and extract 3 pairs, we get 64 types of compensatory base pairs, and 20 amino acids can be specified. How to produce strands for replication of DNA. How to synthesize lagging strand by base pair units. Molecules of liquid water and amino acids have a distorted tetrahedral structure, and they tend to form a helical structure. In the double helix structure of DNA, the atomic arrangement of the constituent helical molecules is heterochirality. Therefore, the 2 do not mix due to chirality, and one becomes the replicational mold of the other. All biochemical reactions are digital. DNA replication is achieved through repetitive operations, similar to the CPU of a computer. Activities of a life through replication of proteins. The signal of start or stop of the work of producing DNA. As the protein lengthens, so does the mRNA nucleotide sequence. When ribosomes are producing proteins according to long mRNA, they also need to produce other proteins. Therefore, ribosomes are produced for each protein. Evolution on replication of protein. The proteins produced by attaching to the cell membrane make the membrane stronger. In addition, amino acids with R groups attached to them along the chain reaction that occurs in the cell membrane form protein molecules and become enzymes. Early evolution of organisms. Organisms that were born by producing proteins have evolved a mechanism to replicate the proteins that sustain the organisms. Summary. Molecules of liquid water and amino acids have a distorted tetrahedral structure, and they tend to form a helical structure. In the double helix structure of DNA, the atomic arrangement of the constituent helical molecules is heterochirality. Therefore, the 2 do not mix due to chirality, and one becomes the mold of the other. Because all biochemical reactions are digital, DNA replication is achieved through repetitive operations, similar to the CPU of a computer. Thank you for your attention.